Procedure of DNA extraction by organic method. In case of organic method of the DNA extraction, the first step is cell lysis and washing. Cell lysis buffer is non-organic detergent designed to lyse the outer membranes of the blood cells. So outer membranes of the blood cells, they are lysed or they are broken with the help of cell lysis buffer. But this buffer do not break down the nuclear membranes. The washing of the blood sample is usually performed with the help of a buffer. The name of the buffer is TE. So TE buffer is used once there is a lysis of the membranes of the cells which are present in the blood. Then the next step is washing of the blood samples to remove those cells that has been broken. So this washing step is performed in 2 or 3 steps. Washing can be in 2 steps and it can be in 3 steps. So at the end of washing, we are left with the nuclei of white blood cells. So at step 2, proteinase K is added, that is for to digest those proteins which are present with the DNA. So these are the proteolytic enzymes such as pronase or proteinase K. So these enzymes, they are used to digest the proteins which are present with the DNA. So they are active against broad spectrum of native proteins which are present with the DNA. So this is the second step during the purification of the DNA when we use organic method. Proteinase K is approximately 10-fold more active on denatured protein. If the proteins they are denatured, then the activity of the proteinase K enhances. So proteins, they can be denatured either by using SDS or by heating. So proteins which are present with the DNA, they are digested by the enzyme. The name of the enzyme is proteinase K, and when we add solution of SDS or we heat the proteins or the solution, it enhances the denaturation of the proteins or the functioning of the enzyme, that is proteinase K. So in case of organic method, there are 2 steps. The first step is lysis of the membranes of the cells which are present in the blood, and the second step is digestion of the proteins which are present along with the DNA.